historically, coronaviruses, vaccines for coronaviruses have had a terrible safety record. There is a condition known as disease enhancement due to pathogenic priming. And this was discovered in vaccinated animals in past vaccine safety studies when they did conduct vaccine safety studies on animals on coronavirus vaccines, where vaccinated animals got more serious disease after being vaccinated, and then when they acquired an infection from the wild-type vaccine, more of animals got serious infections, serious conditions, and more animals died. In my peer-reviewed research paid for the citizens of the United States of America through donations to IPAC, prior to the development of any COVID vaccines, I found that all but one of the proteins in the SARS-CoV-2 virus have what we call unsafe epitopes, right? Which are parts of proteins that are capable of causing immune conditions, autoimmune conditions, and immune responses against proteins in our own body. This is peer-reviewed research, and I'll be happy to provide the press with a reference after this. About a third of the proteins that might be targeted by autoimmune conditions by SARS-CoV-2 viral proteins, either through infection or injection, target the immune system. But from the start, it looks like this virus has evolved the ability to attack our immune system as part of its disease-causing capacity. This pathogenic priming, I predicted in my study which parts of the human body would likely be most affected by pathogenic priming. And now studies by medical physicians, by medical teams around the world are confirming my predictions. This is not simply a respiratory virus with respiratory symptoms. This affects many tissues across the body, and there's some grave concern if you're going to get infected. But where's the grave concern if you're going to be injected with these same proteins? Not a single, to my knowledge, not a single vaccine manufacturer took heed of my warning to remove those unsafe epitopes from the vaccines before they formulated their vaccines, in spite of being emailed my study with a plea to please consider taking out those unsafe epitopes. Your body has 500,000 peptides throughout your life. The coronavirus has something like 28 proteins. What's the probability that there's going to be many proteins that match at small segments of immunogenic epitopes? Very high. We now have results from front-runner vaccines from these hybrid trials where they skipped over the animals trials. FDA, in it absolutely ignoring safety margin in a stunning decision, decided to allow Moderna and other vaccine manufacturers to skip the most important step in vaccine safety for coronavirus, which is the animal trials, to see if there's pathogenic priming, to see if this particular vaccine or that particular vaccine might cause serious problems through pathogenic priming. It's unbelievable that the FDA made this decision, and we made this specific recommendation that they do that in my study that I published. All right. Now what they're doing is they even said, well, we don't have to do phase 1 human trials and phase 2 human trials separate. We're going to combine them together because this is such a horrible epidemic. We need to get the science done fast. They're missing out on another opportunity to find safety signals, but they found them anyway. At the beginning of this time period where I'm speaking, I said to remember the percentage 21%. The only single data point, the datum that we have on what percentage of patients human patients exposed to a coronavirus vaccine have had serious adverse events is from the Moderna trial, and that number is 21%. 21% of people are having serious adverse events from this vaccine in that trial. The other ones haven't published their data yet to the point where we can understand that. Like Dr. New said, they're supposed to be transparent.